I am Dr. KBRS Visarada. I have been serving in ICA since 1986. Since then, I have been working on two important aspects. One is on transgenic sorghum, and the other one is on wide hybridization of sorghum. In the area of transgenic production, we work towards developing resistance in sorghum for sorghum stem borer, that is Chylopatulus, through introduction of Bt genes. And for the resistance, we have used in fact two Bt genes. They are Cry1AA and Cry1B. And these two genes we have introduced into the parental lines that are very popular in India. These parental lines are 296 and CS3541. And we have introduced the two Bt genes, Cry1AA and Cry1B, separately into these two lines with a future view that these two can be combined in a hybrid program. A hybrid can be made a combination of Cry1AA and Cry1B when these two parents are crossed to make a dual purpose hybrid. With the help of my colleague Dr. Padmaja, we have done insect bioassays in which we have tested whether these genes are able to impart resistance in the transgenic lines, and in the process we have identified transgenic lines that have been very good for resistance to stem borer in type 296B and CS3541. To sweet sorghum is all the more susceptible to stem borer because of its sweet juice, and these sweet sorghum lines wanted more resistance for stem borer, and then we selected two parental lines, SSV84 and RSSV9. Using our transformation technology, we have used Cry1A and Cry1B genes, and we also have created transgenic lines and found lines that are resistant to stem borer. And insect bioassays, they were very promising not only for leaf damage but the larval survival also was minimum with these lines. And with the help of my colleague Dr. Shyam Prasad, we have shown the evidence that these transgenic lines are very good for imparting resistance to sorghum stem borer. The third area of research is wide hybridization. So there are species like we have sorghum australians, sorghum purpureosericium, sorghum versicolor. These three species have been recorded for resistance to shoot fly. There are other species, sorghum halipens, sorghum stipodium, sorghum usumbrens. All these species also have been widely grown. Our objective was to bring in those important genes into the cultivated gene pool. The major hurdle in this program is that the wild species are not crossable with the cultivated species of sorghum. So the problem was identified long back. We were successful in overcoming the fertilization barriers between cultivated sorghum and the wild sorghum. And with the help of this pollen pre-treatment method, we could obtain the F1 hybrid embryos with many species like sorghum usumbrens, sorghum australians, sorghum purpureosericium, sorghum versicolor, and many other species. So then these F1s, when we have raised them in the pots, they did carry both the genes of the wild as well as the cultivated parents, and which could be very well seen in F2 generation segregating. One more research area of interest is that we started testing crosses between incompatible species like sorghum and maize. Both are incompatible with each other, so we wanted to study how the crossing looks like. We had seed set in some of the lines, and many seeds were shriveled and deformed and all. Some of the F1s have grown, but we made around 60,000 crosses, a very large number of crosses. Three lines we found the F2 is segregating to a wide range. These progenies we have advanced them through single plant selections, and some of the traits like sweet sorghum with high Brix and high biomass and high juice quality have been identified, and they have been tested in all India coordinated trials, and it has been found very promising. Apart from that, we have forage lines with low HCN content. We identified 2 lines that have low HCN as well as high in vitro digestibility. These have been tested at all India level, found promising, and we recently got registration of germplasm of one of these lines, and these we are making crosses with our elite forage lines. I have been an elected member of Plant Tissue Culture Association.